We have begun recording, so thank you everyone again for coming to see Dr. Valentine. So Dr. Valentine, whenever you're ready.
Okay, well, thank you all for coming. There are a lot more of you than I expected. My name is Kristen Valentine. I have a PhD in quantitative and systems biology, but basically I'm an immunologist, and I have been an immunologist since I started grad school, 6 years ago now. I am now a postdoc researcher in the Shrestha lab at the La Jolla Institute for Immunology. And today I'm going to talk to you about a little bit of my research from my PhD and then how that transitioned into my research now in my postdoc, which is in SARS-CoV-2. All right. So I started my science career in, let me see if I can, there we go, in Merced, which is here in this red star. I was there as an undergrad, and I originally started as pre-med. I really wanted to be a doctor for about 2 years. And then I realized I don't do well with blood or sick people. So I started to try and figure out, how can I still do medical research or medicine without being a doctor? Is there something that I would enjoy more? And in my junior year of college, I took an elective immunology course with Dr. Katrina Hoyer, and I completely fell in love. I absolutely adored immunology, and it took me about a month to ask to join her lab as a PhD candidate. Now, before this, I had been doing some research, not quite like you guys have already in high school, but through college. So I was pretty confident that I liked research, and I just decided to go with my gut and run with it. And through the 5 years of my PhD, I discovered with my lab a new population of CD8 T cells that promotes autoimmune disease. To get an understanding of what that means, some of you may already be familiar with the immune system, but the white blood cells that make up your immune system are actually composed of a ton of different types of cells. One branch is the innate immune branch, and this has things like macrophages and dendritic cells. These cells respond immediately within hours to days right after you're infected or after tissue is damaged. These cells can cause things like fever and inflammation. They're responsible for directly eating things the moment you're infected. They then translate that information from whatever the pathogen is over to these T cell types and B cells. And these cells together are the adaptive immune response. These cells take 7 to 10 days to actually become activated, but they're so potent that it only takes another couple of days after they're activated to completely clear infections. B cells are the things that create antibodies, and now these antibodies are actually what do the function for B cells. Mostly, antibodies can coat the entire surface of a pathogen and completely prevent it from doing any function. Whereas CD8 T cells here, these cells can directly target and kill cancer cells or infected cells. And CD4 T cells are more like helpers. They have a lot of function, but it's mostly to help each other cell type do their job better. My focus for my entire career has been mostly on T cells. I want to know, how do they work? How do they work in infection? How do they work in vaccines? And for the first part of my work, how do they work in autoimmune disease? Now, autoimmune disease is when your immune cells, any one of these, attack and destroy normal body tissue instead. This can be things like type 1 diabetes or rheumatoid arthritis. There's hundreds of different kinds. In my PhD, we mostly studied lupus-like disease. And here I'm showing you an image of a normal, healthy immune response in which the B cells are coded in red or blue, and the CD8 T cells are in green. And what you can see is that in real tissue, these cells are kept very separate from one another. They have very different jobs, they do very different things. They don't need the interaction. However, when we look at the autoimmune tissue here, you can clearly see that there are green CD8 T cells inside of this B cell area. When we observed this, we asked, what exactly are these cells doing? In lupus and lupus-like diseases like the one I study, B cells are absolutely essential for disease. You get rid of B cells, you get rid of disease. So what are the T cells doing in the B cell zone? they're not killing the B cells because you still have them and you still have disease. So instead, we asked what they could be doing. Now, this next phase of research took 3.5 years and I think cost over $50,000, although I don't know for sure and I don't want to know because it was mostly me making mistakes that cost so much money. And what we did is we took different kinds of T cells. We took normal CD4 T cells, which are known to help B cell responses, or autoimmune CD4 T cells, or autoimmune CD8 T cells, and we put them with plain old B cells. Then we looked into see what kind of antibodies these B cells were producing. And what we see is that the B cells by themselves produce a fair amount of antibody. But when they're added with either CD4s that are normal or CD4s that are from the autoimmune setting, you get way more antibody. So the CD4 T cells are doing their job and they're helping B cells produce antibody. But when we add the CD8 T cells from the autoimmune setting, these cells also promote way more antibody. This feature, where CD8 T cells can directly cause B cells to produce antibody, had not yet been seen before in autoimmune disease. Since this paper was published in 2018, there's been a couple more, mostly looking in the situations of infection or cancer that see a similar CD8 T cell feature. But we don't totally know what these cells are doing. So we were left with quite a few more questions. Again, this took about 4 years of my PhD. And we were left to ask, what do these CD8 T cells use to change B cell function? Where do they come from? It's obviously not normal since they're not in a normal response. Can they be found in other autoimmune diseases or infections? And then most importantly, for what I'm interested in, can we use them for therapeutics? In the last year of my PhD, I started asking these first 3 questions, and we're pretty close to getting another paper out for that. But before I could finish it, I graduated. And I left all of that work for the other PhDs in my lab to finish up and hopefully continue working. That's probably the hardest part of any project in science, is that they're never done. There's never a finish date, there's just the time you choose to be done. And I chose to take everything I knew about CD8 T cells and move all the way down here to San Diego, where it is currently 90 degrees and really hot, to study T cell responses in infection at the La Jolla Institute. So I had not originally intended to look at coronaviruses. I joined this lab to look at dengue and Zika viruses, but the pandemic happened, as I'm sure everyone is aware, and me, like so many others, have completely switched gears, and in the last 3 months, I have focused predominantly on SARS-CoV-2. And from what I've heard from Rob, I believe most of your research projects have also started to look at SARS-CoV-2. So, briefly, and hopefully you're familiar with some of this, when we're looking at coronavirus and how viruses interact with the immune system, you have to have a foundation of virology. You need to know how the virus works. So, coronavirus SARS-CoV-2, like all others, is an envelope-positive single-stranded RNA. This means that this one strand of RNA here encodes all of the proteins that the virus uses at once, and different regions of the virus allow it to be continuously produced. This RNA can then be translated directly into protein, which will then form the entire protein. The entire virus. Different proteins are structural versus non-structural. Structural proteins that this virus really uses are the spike protein S, the envelope protein E, and the membrane protein. All of these are on the surface of the virions. It also has a nucleocaspid, which holds the single-stranded RNA, because single-stranded RNA is really unstable. You need something to coat it to keep it protected and organized, and that's the job of the nucleocaspid. All of these other numbers, though, are non-structural proteins, which have different functions for the virus. Some of them are anti-immune response proteins. Some of them are designed to help form the protein. Each one has a specific function, and not all of those functions have been established yet. What we know about how the SARS-CoV-2 virus interacts with the T cells and the B cell responses from humans came about in the last few months, and a lot of those ideas were funded or founded off of what we knew about the original SARS virus and MERS. And most of these studies have involved hospitalized patients. And that's a real strength because we get to see a good portion of the immune response, but it's also a downside because you're only looking at patients that were severely affected. But what we think we know is that antibodies primarily develop against S protein. And this makes sense because the S protein is huge and it coats most of the surface, which is what antibodies would see. But T cells, in a recent paper published by a different lab at the La Jolla Institute, have shown to target S, M, and N equally, and a little bit of the non-structural proteins, although which ones exactly we're not sure. And surprisingly, despite what we would expect about CD8 T cells being highly virulent, CD4 T cells are more important. So what I want to know in my continuing research is exactly which CD4 T cell is most important. Do you need S? Do you need M or N? Do you need all three? Can we pick and choose? To really begin looking at this, and I'm going to show you a piece of data that we just got very shortly, we have to understand how T cells respond to the virus. They don't see the large structure like an antibody would. They instead see little epitopes, which are linear portions of these proteins. So a dendritic cell will eat the virus, cleave it into lots of tiny little pieces that are 8 to 10 amino acids in length, and then show just this little piece to a T cell. So what we did in the lab is we took the S protein from SARS-CoV-2, we cleaved it into little pieces ourselves, and we showed it to CD4 T cells either from the spleen or from the lung. So here you have numbered the different peptides that we use, the different portions of S. We have a positive control, which just shows that our assay worked, and a negative control, which tells us what the lowest response we care about is. And now spot-forming cells just mean the number of cells that actually responded and did something compared to the positive control. So against S protein, epitope 2, 3, 4, 6, and 7 all had significant responses compared to the negative control. So these particular regions of the S protein are more important to mount a CD4 T cell response. But what surprised us is that 2 and 4, for example, have very different responses between the spleen and the lung. We don't exactly know what that means yet, but potentially we could use this information to create maybe a vaccine that's directly targeted to the lung versus the spleen. Now we're going to try to continue to do exactly the same experiment for all of these other regions. We tried N protein in the same way as we did the S protein this week, and it completely exploded. I mean, nothing worked. Everything read 0, which probably means my assay didn't work and I have to try it again. But it could also mean that there just aren't T cell responses to N. But that would contradict what everybody else says, so I'm pretty sure it's my fault. So what I'm hoping to do is, as I said, redo this same experiment against all these other proteins and evaluate which ones are the most important in an immune response, and then take those specific T cell responses and see how long they last both in humans and in other infection models to see if we create memory. Can we make memory last for months, years versus days so that people can be protected for a longer period of time to SARS-CoV-2? And can then we use that information to create a vaccine target that will produce a really great response that even if you get infected in your lungs, it's completely cleared before it gets anywhere else. So my research will continue to focus on T cell responses and vaccine models. Other members of the lab are starting to look at the innate immune response and understand how those work. So my last slide is just to thank the people in my current lab. Most of my work has been done with another postdoc, Angel and Annie, and everyone else has been super supportive. Unfortunately, all of our undergraduate interns who are doing research aren't allowed to come into lab until this is all done. But we hope to create some really interesting SARS-CoV-2 vaccine candidates to look at in the future and hopefully get past this pandemic. And I am happy to take any questions on any of the aspects that we talked about. Thank you so much for having me talk.
Thank you so much, Dr. Valentine. That was a wonderful presentation. Does anyone have any questions? You can either put it on the chat board or vocally say anything. So there was one question. Dr.
Yes.
Valentine, do you mind going back to the slide where you were talking about the assay with the different, yeah,
This
this
one?
one here? Yeah,
Uh-huh.
someone wanted an explanation on the chart you have on the right, just to explain what's
This
going
one?
on. Yeah.
So this is a diagram from a review, which this is the citation here. And what this is showing is all of the different pieces. If I go back one more, well, here. So it's basically showing you how this entire region of SARS-CoV-2, which is the whole RNA, gets broken down into different protein subunits that are then used to create the actual virion. So the S protein is the longest, then there's 3A, which is cleaved in different ways. So the green portion here is the actual protein that's used, and the gray portion is the remaining stuff that will get cleaved off by the cell when you're creating the new virus. Does that make sense? Hope so. Ask another question if it doesn't make sense. Okay, somebody asked, what got you interested in immunology specifically? So I was sitting in class, and it was like 8 a.m. class. It was really hard to stay awake. I drink a lot of coffee in college. I still do. And my teacher talked about how a single CD4 T cell, of which you have over 1 million different CD4 T cells in your body, is responsible for every response you have. So one cell will respond to one pathogen, and the rest of the CD4 T cells that you have are irrelevant. And I asked her at the end of class, because I was really shy and I didn't want to speak up in class, how does a CD4 T cell, if there's only one in millions, how does a CD4 T cell find the virus? And she said, we don't know, not fully. And that was kind of it. I wanted to know how. I wanted to know why. I wanted to know why some people get sick way worse than others, why some gentlemen complain more about getting the cold than some ladies. I wanted to understand what aspects of the immune response drive how we feel and what we view, and if we can make them better, because it's such a potent tool in medicine, and we have almost no idea how it works, even though it's been around for hundreds of years. So I kind of just took it and ran, and I've been really lucky because I still love it. Okay.
Dr. Valentine, the next question we have is, so one student says, I have heard a lot about the virus, how the virus mutates very fast. So will your vaccine be future-proof in a sense?
Potentially. So not all of the mutations on the virus are as important as each other. Some cause the virus to be more infective. Some cause the virus to be potentially able to evade the immune system better. We don't exactly know how each mutation works with the virus. In theory, the vaccines that we're hoping to create will withstand those different mutations and be able to work around them. So even if one part of S gets mutated, hopefully the other parts that we're also targeting will still be just as potent and be able to get around that single mutation.
Awesome. We have another question that says, did you by chance study the coagulation cascade that is triggered by the binding of the virus to the ACE2 receptors?
That's a great assay, but we aren't doing that style of assay yet in the lab. So no is the short answer. The La Jolla Institute is working on creating a BSL-3 facility so that we can work with the live virus. Most of the coagulation cascades and ACE2 receptors are still using pseudovirions, and we're hoping to use the live virus instead, but we're waiting on the facility to be finished for us to start looking at those different aspects. Maybe biasly, and maybe because we have a BSL-3 coming, but we think that the live virus assays will give us more information than the in vitro assays using pseudovirions, but also ACE2, pseudo-ACE2.
Awesome. And a follow-up question to that, the same student asked, is this a big reason on why so many COVID patients are dying of blood clots, or do you think there's another potential reason that that could be the case?
Maybe. So ACE2 and the importance of ACE2 on the surface of different cells is understudied. And it's a good starting point to try and understand what the virus is actually doing to people who are dying. But the caveat to that is that we don't actually know which part of the response is killing patients. Is it the virus and something related to ACE2 and coagulation? Or is it something like a cytokine storm, and that's completely separate, and it's based on the immune system? In either situation, you can get things like blood clots, you can get things like sticky cells coming together. One other aspect that can cause coagulation is also antibodies. If you get enough antibodies in a system, they can cause things to stick together more easily and also form clots. So I think that patients that are dying of blood clots, it could be one of three things. Right now, the evidence that I have seen has suggested it's cytokine storm and not viral related, but I'm not sure we know yet, is the short answer to your question.
Awesome. Dr. Valentine, a student was asking about your experience with PhD. Is it something you would recommend, or how would a student go about, learning more about the PhD lifestyle in itself?
Sure. So a PhD is excruciatingly difficult, no matter which PhD style you choose. However, it is not more difficult than any other graduate degree, MD, pharmacy, anything like that. That being said, it presents some unique challenges. The hardest thing for me in a PhD to do was learn how to fail over and over and over again and still continue to ask questions and not take that failure personally. I don't like failing. I don't think anybody does. But the process of a PhD, while really rigorous, is different than school. It's different than even going to college. You don't have to do anything. You're choosing to ask the question because you enjoy it, you like it. So every time it gets difficult, it's something you really love to do and you really enjoy it. That being said, I had a lot of really great friends and really great support, which made it a lot more fun. And I think a lot of people have really negative experiences with PhDs because it's ambiguous. How to succeed in a PhD, your entire project can fail for 5 years and you can still get a PhD. And that's hard when you're trained to take a test every day, and that's the measure of success. So in terms of the second part of that question, how to get the PhD experience, when you're in college, and even now, if you have the opportunity to volunteer in a research lab and work with PhDs, that's what really started to teach me about what a PhD would be like. And even that fell short of the actual PhD experience. But don't be afraid to ask. Go when you're in college, or even now, if you know of somebody doing research, ask if you can volunteer in their lab. It will be extra work on top of your already busy workload, but that's what a PhD is. It's getting into the lab every day and doing research and experiencing the failures and the triumphs and kind of getting through it at some times and really enjoying it at others. And I hope that actually answers your question. But
yeah, that was great. And I think maybe, the failure aspect, especially, I think a lot of students are, programmed to hate failure, but a lot of times failure is what makes someone grow. So I thought that was a great point you made. So can you explain why SARS-CoV-2 antibodies are not being used as a treatment as of now?
So they are being used as testing all over the place. As of about a month ago, there were 1,427 different experimental treatments in the clinic. And that was everything from stage 1, which is like in a mouse, to stage 4, which is in a human. And about a third of those were focused on the idea of using antibodies to treat patients, convalescent antibodies. So patients from a convalescent patient antibodies into other people. There are two potential issues with using antibodies. Well, 3. 1, they may not work very well. If you look at the patients who are dying of SARS-CoV-2, they're all mounting an immune response. There are very few people who are having negative effects from SARS-CoV-2 that didn't have an immune response. So the immune system's working, it's just not working efficiently. So if you get inefficient antibodies and you give those to people, they're still not going to be protected. There is also the idea that antibodies, if they're there and they're not super strong, that the virus could use the antibody to invade other cells. And that's a process called antibody-dependent enhancement. There's very little evidence to suggest that that can happen, but that's a risk with all viruses and all antibodies. It happens with dengue and Zika, so people are worried that it could with SARS-CoV-2, and the last thing we want to do is make it worse. And then the last reason is that antibodies from humans only last for a very short amount of time. Antibodies, like all proteins, have a half-life, and you can't immunize the entire world at the same time with enough antibody that we would eradicate SARS-CoV-2. So all you would be able to do is put a stopgap and maybe, possibly, and what they're hoping, save some of the very, very worse off patients, but it won't work as a vaccine, maybe only as a treatment. And even then, it may not last long enough.
Awesome. And I think that's like a follow-up to the next question. Do you think the vaccine for COVID would be like the vaccine for the flu, where there is a new one each flu season just because of the mutations, or do you expect it just to be one overarching vaccine?
So the mutation rate of coronaviruses as a general rule is actually really low compared to something like the flu. The flu mutates like crazy, and even though we're seeing coronavirus mutations, it actually mutates less than the flu. The fact that we're seeing so many in SARS-CoV-2 is likely because it's going through so many different people so quickly. That being said, I don't know if we can even begin to estimate how a vaccine would work because we don't have one that works yet. It is possible that people will try to model the future mutations and create vaccines for the future, but we've had so little success creating an effective, safe vaccine that the idea of having one that we'll then have to roll over and over and over, it's possible. The more relevant point that I think people, at least in the institute that I'm at, are considering is actually, can we create a vaccine to different coronaviruses that already exist? If we can do that, then maybe that vaccine will be robust enough to not need to make more over and over because we won't have to worry about the mutations. But so far, no one's been successful, at least as far as I know. Could be wrong.
Awesome. And, you know, I think you bring up a good point with the different types of SARS-CoV, like there was SARS-CoV-1 and MERS. So can you sort of mention, you know, why is it that the infection rate is so different between, you know, SARS-CoV-2 or SARS-CoV-1 and MERS, and, you know, maybe structure or functionally, what makes them so different?
So I can't speak to the structure or function. For those of you who are really structurally and functionally inclined, if you're willing to wade through some heavy duty reading, the Sapphire Lab at the La Jolla Institute is a biochemistry structural biology lab that used to work in Ebola and structural functions and differences in Ebola, and they're now doing SARS-CoV-2, and they have a paper coming out in the next few weeks. But that may start to address some of those questions. In terms of the infectivity, we know that SARS and MERS infect similar amounts of people that come into contact. However, there's something like super spreaders are thought to be much less for SARS and MERS, and you're also thought that you have to be experiencing symptoms to spread SARS and MERS, whereas SARS-CoV-2, we're starting to believe and seeing more evidence that asymptomatic people are also spreading it. So there's a little bit of the ease of spread and probably the amount that is spread when you cough. So SARS may not actually have as much in a cough as SARS-CoV-2. The one other aspect structurally that I just thought of is the structure of the S protein is different between SARS-CoV-2 and SARS. And it looks like in early studies that the spike protein of SARS-CoV-2 requires less effort to break, and you need a break to happen inside of it. It's actually a cleavage that happens in S to allow it to invade a cell. And that looks like it's easier for SARS-CoV-2 than SARS. So that may be some of the differences that we've seen between the different styles of coronavirus. But a lot of the research is really cutting edge. Everything we're finding is brand new and we had no idea.
Awesome. And in your lecture, you sort of mentioned that the adaptive immune response takes a longer time to be activated. Is there any potential way to activate it quicker than original? I don't know why I'm getting some reverb, but is there a way to activate it any quicker to target coronavirus, or is that not really a possibility at this point?
So the immediate amount of time is 7 to 10 days, as I stated, to activate the different cells of the adaptive immune response. And that's not just activation. You also have to, as I stated in the idea of the thing that interests me the most in immunology, you have one cell, maybe two, that are responding to the same virus or the same part of the virus. And that single cell has to expand sufficiently that it can actually combat the virus, and then it has to travel there. So most of the expansion occurs in secondary lymphoid organs like the lymph nodes or the tonsils or the spleen. And then wherever the virus actually is, those cells have to get there. But getting there is actually the shorter part. The expansion, T cells can expand, I think it's every, I want to say 18 hours inside of the body. It might be a little faster, it might be a little slower, but somewhere in that range. So you can get a good expansion, but that takes time. So speeding up that time has so far not been possible, at least at the adaptive response. That's where the concept of the vaccine comes in. If we can vaccinate you before you get it, then you have a bolus of cells that are responsive already, just floating around doing nothing, that will expand and respond much faster than the naive population inside of your lungs. And that's what most T-cell vaccines try to do. They're just harder to make than B-cell vaccines.
Okay, awesome. And shifting a little bit back to your educational career, how you got through the PhD. Do you mind commenting, within those PhD years, is it mostly just lab time? Is there classwork? We just had some students sort of curious about, within the PhD, what sort of goes on within those years.
It varies by school. And it depends on which school you want to go to. So if you decide to go for a PhD, one caveat I'm going to tell you right now is, unlike an MD, where you send out one application and it gets sent to all the different schools that you choose, it just costs more money. A PhD, you have to make a different application for every single school you want to go to. That's different letters, it's different essays, it's different requests for every single one. Most people that I know applied for anywhere from 3 to 10 schools, and that was it. And then once you're in, what they require, typically it's the first 2 years you finish any and all coursework that you may have, and the second 2 to 3 years is going to be all of your laboratory work. The first 2 years you still do laboratory work, but you do more of the classwork as well. And then by the time you hit your third year, you should be completely done with classwork, typically. Some schools require that you also have to teach, as in be a TA for students in college. UC Merced at the time required that I taught for 4 of my 5 years. That was more financial than actual requirement. And that takes a lot of time away from the bench. But once you switch over to the bench, you're expected to finish a project or at least demonstrate that you can think about a project. Again, not everything always works, and you can still graduate even if it doesn't work, provided that you really thought your way through it and you can explain why things didn't work the way you expected. Really testing in a PhD is testing to see how you're thinking about the questions and how you're using the resources you have to answer questions. Did I answer your question? I hope I did.
Awesome. Does anyone else have any questions related to what Dr. Valentine talked about? I actually had a question, Dr. Valentine, if you don't mind. I just wanted to hear sort of your perspective on some of the drugs that are being used to target coronavirus, like remdesivir and hydroxychloroquine. Do you have any sort of perspective on, maybe how the media is handling it, or, if this is a safe drug that should be prescribed to patients?
So I think that the downside of all of the work that's being done right now to apply different drugs to different people is that a lot of these drugs haven't been tested in the number of people requiring that they need them. So, for example, I'm trying to think of which drug I want to explain, but we'll just pick one. Drug A is previously used in people who were 19 to 25 to treat acne, we'll say, as a hypothetical. Then we go, oh, maybe it works against a virus, and we apply it to 500 people, anywhere from age 9 to age 78, and all of a sudden we're seeing these weird responses. You can't possibly predict all of the negative repercussions a single drug will have across that large of a population with that many different variabilities. That just accounts for age. We're also talking about variabilities of age and race, as well as available medicines, prior medications, previous conditions. All of those things play a factor. So when you see in the media that a drug failed, what is the failure rate? Is it really failing across the board? Is it really causing these issues across the board? Or is it failing because it hit the 10% mark for complications, and so they're saying we can't use it, which is also still possible. But it may be that it's not failing in general, but failing in specifics. And so if that's happening, then the media is likely just working without all the information, and some of these drugs may still be good in some people. They're just not good in all people. But the new wave of the future, in my opinion, and for drugs as well, should be somewhat personalized medicine. You have to look at the individual, especially when you're talking about modulating the immune system. Each individual person is going to require something different. It's going to need something different. So we won't be able to use blanket drugs. So in short, yes, these drugs are probably not as safe as they had hoped they would be. No, they're probably not as bad as the media is portraying them, but more research is really needed to really create something that is broadly useful. And if it's broadly useful, it's probably not very strong because it can't be. That being said, I think I saw a question in the feed here. When do I think a vaccine will be created? Even if a vaccine is created tomorrow, I'm not sure that I would go out and get it, just because I think I would want it tested more in more people and in more situations. And I'm not sure I want to be the test pig, but who knows? That being, I mean, we all want to get back to normal, but it's going to be at least 6 months, assuming that a drug has been created that works already, for it to be fully tested and out in the market. We've expedited a lot of things to try and combat this, but as you can see, they're not always successful.
Awesome. Yeah, and I think the big thing a lot of times with developing vaccines is that there's so many steps that you have to take that, even if someone says they're going to develop a vaccine in the next couple months, you're going to be hesitant whether they ran through all the clinical trials, got through FDA regulation and all that. So I think that's an awesome point. Sorry, Dr. Valentine, just shifting back, I think we had a couple of lingering questions back to the PhD portion.
Sure.
Would you recommend going from a bachelor's to a PhD as opposed to getting a master's prior?
That's what I did, but it depends on who you ask. So when I went for my PhD, I had extenuating circumstances. I had a lot of student debt that I didn't know how I was going to pay back. I already was pretty confident that I wanted a PhD. I was right. I did, and I'm happy. And I got in, which is the other aspect. It's not a sure thing. A lot of people think of a PhD as, oh, I'll apply for med school. If I don't get in, I'll go get a PhD. Sometimes that works. Sometimes that doesn't work. So in the situation for me, it was great. I didn't waste time. I didn't do a project that only lasted for 2 or 3 years and then have to change again. But it doesn't always work out that way. I think if you're not sure, 100% sure you want a PhD, a master's is a phenomenal way to get that feel for a PhD without committing to a PhD. If you're thinking, I really would rather go to pharmacy school, then maybe a master's instead of a PhD beforehand. If you're thinking, oh, I want to be an MD PhD, just go be an MD. You don't have to be an MD PhD. You can still do research with an MD. Very possible, very good, and you get a great education both ways. So there's lots of other things to consider rather than just go bachelor's to PhD. And then the one other aspect is it's a long haul. You're pretty tired even after a bachelor's. You're tired of being in school, even in school you choose. You work really long hours, and in your bachelor's, you make no money. And in your PhD, you technically get paid, but not very much. So you have to be sure that you want to commit to that and not to do something like take a break or try something different. So for me, it worked. It's not the right recipe for all people. And I have had mentors who are PhDs and who are MD PhDs who did master's first and really enjoyed it. The wrong avenue to take is to say, well, I don't want to be in school till I'm 40. So then skipping a master's. It's better to take the time to be sure, because you can always change your mind and you can always do something different. No choice that you're going to make between right now and the day you die is irreversible. So take the time you need to decide to do the thing you really love. It's also another thing for a master's. It's a good way to try out a science without committing to being in that science. If I had hated immunology, I could have still switched after my PhD, but that would have been a long 5 years doing something I hated. That makes sense.
Awesome. And then you mentioned that a lot of money was spent on mistakes you made in the lab.
Yeah.
Can you sort of elaborate on that and how your mistakes in general, shaped your career?
Sure. So there's a few different ways that mistakes shaped my career. The first one is the personal mistake, where I did not pay attention. And so I made a mistake because I could have been smarter about the way I physically did an experiment. And the experiment failed because I messed up. And in those mistakes, they're very personal and they kind of hurt your feelings because you're at fault and there's no one else to blame. There's nothing unexplained. You made the mistake. And in those, what I got from that, from a really great trainer that I had, a technician and a mentor that worked with me, was to stop and think about what you're doing before you do it. It's really exciting to look at a lab and to just get started, but you should have a plan. And that's true for pretty much anything you ever do. You should stop, think about it, think about your question. Is it the right question? That's a great way to start. And then how do I ask that question? What are the parameters? And what are the things that I need in my experiment to make heads or tails of it? And the second kind of failure is the failure where the experiment, whatever it was, will say, I'm trying to think of an experiment you may do in lab, a tissue culture experiment. Okay, let's say we're doing a tissue culture experiment. The cells grew, but they didn't grow the way I expected them to grow. That's okay if I have a positive and negative control. Now, my boss might not be very happy. She may wanted to see something different. But if I am sure that it worked, then yes, it failed to answer the question I had sought to answer, but it still gave me usable information. So to transition from the personal failures to the usable information failures takes a lot of willpower. It also takes a lot of willpower to stop and say, okay, it failed and my boss is unhappy, but it doesn't matter because it still worked in its own way. Then there's the overarching failure of, I don't want to do this, or I don't want to ask that question, and I've been pursuing this for this long, and somebody else answered it before me, or, I just don't care. Or the concept of, like, we should answer this question, but it's not possible. There's things like that which put roadblocks in your way, and they can be really informative in making you stop and think about what you're doing, why you're doing it, whether or not you could do it in a different way and get creative. But most of the failures just feel like failures, and learning to separate what feels like an F from what is not actually an F is really an important process. And for me, it was really hard. I think some people have an easier time, but I'm really horrible at failing. So I still am. I'm still learning. So to get to that point, it's important. And just take a step back when your feelings are hurt, I think is what I'm learning right now. I recently submitted a paper to my boss and she sent it back and it looked like it was bleeding. There was so much. I thought I was a good writer. I'm still a little hurt about it, but it's part of the process and I'm going to learn from it and I'm going to write something better. I already have written something better. I know it's getting better. So there's things like that.
Awesome. And, it's great that you mentioned that, Dr. Valentine, because someone as accomplished as yourself, it's good to know that you have the same experiences as some of these students might have.
Well, there's one more thing for that. There's also always somebody who's going to be better at it than you are, whatever it is. It could be writing, it could be thinking about a question, it could be doing the experiment. They could be putting physically more hours into it than you are, and so getting more data. It's okay. It doesn't have to be perfect. You have to be happy, and that's a hard thing to define.
Awesome. Does anyone have any last-minute questions? I think, Dr. Valentine, one last question, I guess. Something came up. So we actually had a discussion about COVID last week, and one of the biggest topics that came up was about, the effects of heat on the virus in itself. And I know there's still a big area of research since, summer's is here and we're seeing the infection rates sort of going higher. But do you want to maybe comment on, how maybe temperature can affect coronavirus?
So there's two ways in which people or at least two ways that I know of, that people have been talking about the effects of temperature in SARS-CoV-2. There is the mechanistic structural biology level, which says, a fever is a good thing because it causes the proteins on the virus to break down in different ways. The RNA is not as stable at high temperatures and things like that. And that also translates to surface areas that we are around. So if a benchtop at 50 degrees Fahrenheit can hold a virus, maybe it can't hold that virus for as long or at all if the benchtop is at 100 degrees. So there's that aspect. And in relation to that, I don't know that much. The paper that I read recently was mostly on looking at the structural integrity of SARS-CoV-2, and it found that the spike proteins break down more quickly at higher temperatures. And that could be good because it could cause the virus to be less infectable, or it could be bad because it could cause the virus to be more infective. I'm not sure anybody has figured exactly out which way it's going. The other aspect that people, epidemiologists are talking about is the concept that the infection rate is expected to go down in the summer, just like flu would. And the reason for that is that people are not being cooped up together. They're going to spread out. They're going to get away from each other. They're going to be outdoors instead of indoors. And we already know that the aerosolized form of the virus spreads more quickly indoors than outdoors. The caveat to that is what I have seen, at least at the San Diego beaches, people are not actually staying away from each other. So yes, we're outdoors, but they're still right next to each other. So hopefully, high temperatures means that the virus breaks down more quickly and is less infective. And we'll see if that's actually true. I don't know if anybody actually knows what to expect from this virus.
Awesome. Thank you so much, Dr. Valentine. I think that concludes our Q&A session. Again, I could speak for all the students here. Really appreciate, bringing your expertise into SARS-CoV-2 as, this is such a current problem. So definitely appreciate everything you've told us. One other question that sort of was brought up was, if any student has any other questions, would it be okay to email you?
Absolutely. I think you have the email for my UC Merced account. Feel free to do that. I may not get back to you right away, but I will try. And hopefully in a 6 months time, I should have some new interesting data that I can send to you in, I hope, a paper. Otherwise, I'm happy to give more talks. I had a great time, and good luck to all of you.
Awesome. Thank you so much, Dr. Valentine.
May I ask one last question, if you wouldn't mind, Dr. Valentine?
Sure, go ahead.
What advice would you give for any students who are interested in taking immunology in college?
In college? If you're pre-med, I personally think you must take an immunology class in college. 2 reasons. One is because how do you study the human system without studying its natural mechanisms for handling viruses and damage? That's my own personal bias. The second, for MD perspectives, you will take immunology in med school, and it will be harder. If you give yourself the time in college to tackle these difficult subjects, you will have an easier time with your first round of exams in med school. I don't know after the first round, but I know it's easier for the first round. The second thing is, for those of you who are just interested in immunology, it's a fascinating subject. If you need straight A's to do whatever you're trying to do next, immunology is one of the easiest classes to fail. It's extremely difficult. It takes an absurdly large amount of time. We basically ask you to learn an entirely new language in 2 weeks and then apply it to complex situations. It is well worth it to me, but it takes way more time than 4 classes altogether will allow you. If you choose to take immunology in college at any of the UCs, my suggestion is to take it in a year, in your second to last or last year, when you can take it with 1, maybe 2 other classes of which you feel comfortable in already. You may find that immunology is a breeze for you and you will be the rare person, and I hope it's true and I hope you love it. But it is also, it's very difficult. So don't expect to just walk in and be an expert if you do find that it's difficult. The second half of that answer is, if you choose to take it, the first half of every immunology class is really boring. All they do is make you read the book and learn vocabulary. It feels like you're in English class, except for it's a science class. When you get to the second half, that's when it gets cool. Different instructors, including myself, have tried to teach it in a different way so that it was more engaging all the time. It just doesn't work. So get through the first half, work really, really, really hard, and then it's really cool. That's about the best I can say.
Great. Yeah, so if anyone else has any other questions, I will include Dr. Valentine's email in my email for tomorrow. So you can feel free to email her.
Yes, absolutely.
Again, thank you so much, Dr. Valentine.
Thank you.
Awesome. If all the students can still stay on the call for